Here at the University of British Columbia, 4 new species of red algae called Pyropia have been discovered using molecular DNA sequencing techniques.
I'm Dr. Sandra Lindstrom, and I'm an adjunct professor in the Department of Botany here at UBC, as well as being affiliated with the Beatty Biodiversity Museum.
Dr. Lindstrom has had over 30 years of experience working with red algae. She recently used her experience and knowledge to collect different samples here on the West Coast for her research.
But season is very important because a lot of these are seasonal species, and in fact winter is the best time. Pyropia species occur in the high intertidal, so it's not so critical that you go at low tide.
Once the samples are collected, they are brought back dried in silica gel to the lab. Her lab at the Biosciences building of UBC has the equipment necessary for separating the DNA from the dried samples.
For using gene sequencing for these species in particular is that a lot of them really look alike.
The RBCL gene encodes for protein in the ribosomes, the cell organelles responsible for protein synthesis of the Pyropia cells. This gene is carefully selected from the collected DNA with primers specific to the gene and then placed through the PCR, polymerase chain reaction, which makes for many copies of the target gene for sequencing.
That it is one of the best genes there is for not just looking at species differentiation.
The DNA that Dr. Lindstrom isolated is sequenced on campus. Once the results are returned, Dr. Lindstrom can determine the different species based on the differences in the gene sequence. Finally, the last step is to describe the physical appearances of the newly discovered species and create a key for them. Due to the physical similarities, however, the key is dependent on the genetic information of the samples.